Welcome everybody. Today we're going to be talking about immunity to tumors. And a lot of this we've actually covered a lot before in previous lectures. We've talked about the role of CD8 plus cells, the cytotoxic T lymphocytes, in killing tumors along with virus-infected cells. We've talked about the need for cross-presentation and the display of cancer antigens on class 1 MHCs. We've talked about how cancer cells use inhibitory mechanisms in T lymphocytes, and we've even talked about therapeutic strategies to hinder those inhibitory mechanisms, like with PD-1 and CTLA-4. And now we're going to talk about some of those mechanisms and multiple strategies along those lines a little bit more in detail in this chapter. So let's get into it. First of all, there are a few fundamental traits to tumor antigens and our immune responses against them that are fundamental to understanding tumors and cancer itself, and then also therapeutic strategies against them that we'll talk about later in this chapter. The first trait is that tumor cells stimulate a specific adaptive immune response through a number of different branches, not just CD8 plus cells, that has the potential to prevent or limit the growth and spread of cancers. So two takeaways from that. One is that, yes, our body is designed to fight these. Second point is that I always have potential because immune responses frequently fail to prevent the growth of tumors. And because of that, because of those ineffective adaptive immune responses, we've developed ways to engineer and design strategies that can re-stimulate and augment our own body's responses. And that's really what we want to focus in on, particularly in the second half of this lecture. Now, first, there's just a few terminology that I want to take a time out and describe. The first one being neoantigens, which are, in theory, self-antigens that develop only on unhealthy tumor cells. And what's important about this is the fact that they develop later on in life and in the peripheral or past the peripheral lymphoid tissues is that our body has not developed tolerance against it. So yes, despite the fact that they are self-antigens, there's no tolerance mechanisms to them, which is why the immune system can fight it off against it and why we want it to fight off against it as well. We also talk a little bit about genetics in this particular chapter, so I want to bring up the term oncogene, onco being one of those Latin or Greek root words that designate or designated for cancer. And oncogene is a gene that has the potential to, when it mutates, cause cancer just by the nature of whatever that gene that it encodes. So any gene that encodes mechanisms for hindering apoptosis or augmenting proliferation strategies like receptors for growth factors are potential oncogenes. So one for epidermal growth factors is a common one. One's for VEGF, so receptors that recognize VEGF. Also the gene that encodes the growth factor receptor for HER2, which is commonly seen in breast cancers. Now, ultimately, cancer-fighting lymphocytes target tumor antigens, and there's a number of different sources to where these tumor antigens can arise. The first one we just previously touched on is the term neoantigens. And again, neoantigens are self-antigens that derive from mutated oncogenes in tumor cell clones. Now, they themselves can be classified into two different categories. There are passenger mutations, which are by far the most common forms of neoantigens, which are simply just due to the product of randomly mutated genes and have nothing to do with the actual apoptotic mechanisms themselves. Whereas driver mutations are ones that are directly the result of the mutated oncogenes. So they are required in order to drive further cancer proliferation. The passenger mutations are simply a byproduct of the highly mutagenic nature of cancer cells, because they can have a lot of mutations going on due to their rapid proliferation and actual rapid dying as well. So a lot of mutations can occur that have nothing to do with the cell proliferation cycle, which is why these mutations are simply passengers in the sort of trip that is cancer, as opposed to the actual drivers of cancer and the rapid proliferation themselves. So that's what differentiates those two. There's also a number of viruses that are implicated in the development of tumors, and they produce byproducts themselves that aren't endogenous or innate at all. So they're not self-antigens, they're completely foreign. They're byproducts of the virus that find themselves in the cytosol or perhaps cell membrane of these now mutated tumor antigens. Some common viruses that are implicated in the development of tumors are the Epstein-Barr virus, which is linked to B-cell lymphoma, and the human papillomavirus, HPV, which is linked to cervical cancer and other types of cancers as well. There's also other antigens that are present in otherwise healthy cells, except they're just overexpressed in tumor cells. Some great examples are just some of the ones previously described related to oncogenes that are receptors for growth factors necessary for the rapidly proliferating tumor cells that are overexpressed in those cells then, as opposed to healthy cells that have nowhere near that need for proliferation. So they wouldn't have that many growth factor receptors, for instance. Now, this is actually a little bit puzzling because neoantigens and antigens of oncogenic viruses, it's certainly understand why we might develop immunogenicity against those because we wouldn't have developed tolerance or they're foreign. Whereas these are healthy self proteins that we would have developed a tolerance for. But so there's a few different theories why they can still be immunogenic and targeted by our own immune system. One is that that antigen might not normally be produced except in only early developmental cycles of a particular person. So we wouldn't have developed long-term tolerance for these type of proteins. Or maybe they are present even in healthy cells at all times, at all stages of life, except their excess concentrations have a way of activating lymphocytes, whereas their lower concentrations present in healthy cells wouldn't be sufficient enough to overcome our own body's inhibitory mechanisms to activate lymphocytes that are reactive against them. Now, perhaps this slide needs no introduction, but I'm going to give one anyways. But the principal mechanism for immune protection against tumors is killing of the tumor cells by CD8 plus cytotoxic T lymphocytes, the same way we get rid of most virus-infected cells. There was a lot of early evidence that directed us towards this understanding. First of all, is that tumor antigens present themselves on class 1 MHCs. So even though we know that CD4 plus cells are going to be indirectly related to tumor immunology, the most direct way of killing cells would have to be CD8 plus cells, because now we understand CD8 plus cells are linked to class 1 MHCs. Second of all, in that host with established tumors, we've been able to isolate cytotoxic T lymphocytes and have found that they are absolutely specific towards those types of tumors, so they won't be present otherwise. In all cases, perhaps a really good indirect way of understanding this principle is that a prognosis against cancer always correlates with increased cytotoxic T lymphocyte concentration. So if you know you have a high concentration of your CTLs, you have a good prognosis. If you don't, well, then you're not going to have a good prognosis. Really sorry to hear about that. Now, in multiple chapters, we've talked about cross-presentation, which is the ability of dendritic cells to sort of ingest. Previously, we talked about virus-infected cells, so that the dendritic cells, which are necessary to activate T lymphocytes, can sort of digest those antigens and let them slip into the cytosol so they can display them on class 1 MHCs. We know this is necessary because dendritic cells are the most effective way of activating CD8 plus cells. So in tumor responses, cross-presentation is almost always necessary because it's just statistically improbable that the dendritic cells are going to be the ones of the original mutation for cancer. So tumors rarely start on antigen-presenting cells. Also, dendritic cells are just really good of expressing all the necessary co-stimulators. So most cells don't have this. So they wouldn't be able to activate CD8 plus cells initially the same way dendritic cells can. And lastly, it should come to no surprise that CD4 plus cells do indeed offer assistance to CD8 plus cells. So other cells don't express the class 2 MHCs in the same way that professional APCs like dendritic cells do. So in order to get the full response, cross-presentation has to occur and dendritic cells have to activate CD8 plus cells initially to fight against cancer. Now, as I mentioned, CD4 plus cells do offer assistance to CD8 plus cells. As we learned in many previous chapters, there's almost no immune response that acts totally independent from other responses. They're all somehow interlinked, and there's ways that the innate and adaptive immune responses augment each other in different ways and vice versa. And this is no different. The CD4 plus helper cells, particularly Th1 subtype, can do this through a number of different mechanisms. They enhance CD8 plus T cell responses. They activate macrophages that secrete tumor necrosis factor, TNF. They produce IFN gamma, which increases the expression of the class 1 MHCs that are necessary to present tumor antigens. And also that cytokine increases the sensitivity to lysis by CTLs. Now, because I'm not the most eloquent person describing this, I'm going to let this little animation help describe exactly what I was just trying to get across. Hope you enjoy it.
CD8 T cell responses to tumors may be induced by cross-priming or cross-presentation, in which the tumor cells or tumor antigens or both are taken up by dendritic cells processed and presented to T cells. In some cases, co-stimulators expressed by these antigen-presenting cells provide the second signals for the differentiation of the CD8 T cells. The antigen-presenting cells may also stimulate CD4 helper T cells, which may provide signals for CTL development. Differentiated CTLs kill tumor cells without a requirement for co-stimulation or T cell help.
Fascinating. Just fascinating. Now, moving on from CD8 plus cells, our body has other immune response mechanisms that help fight against tumors. The first being through natural killer cells. We certainly have evidence of that in that there are many studies that support the role of natural killer cells against fighting cancer. So, for instance, we know that genetic defects in the development of natural killer cells correlates with higher risk for developing tumors. And there's a number of different mechanisms that help explain the natural killer cell's role. And the first one we've actually talked about before because viruses do this too. They'll downregulate class 1 MHCs as sort of a way of escaping recognition of the normally tumor or virus-fighting CD8 plus cells. Now, the class 1 MHCs are a normal inhibitory ligand against their cytotoxic activity. So by downregulating this, yes, they evade CD8 plus cells, but then they just invite more augmented responses by natural killer cells. Also, natural killer cells themselves have some sort of co-activators or co-stimulators that help initiate their cell lysing, their cell-fighting responses. Tumor cells will actually express ligands for these activating receptors like the NKG2D receptor on natural killer cells, which act to override their inhibitory signalings whenever they recognize class 1 MHCs. So even if a natural killer cell does see a class 1 MHC, the overexpression of these ligands for their activating receptors will override that, cause them killing that cell. And lastly, antibodies will activate through antibody-dependent cell toxicity whenever anti-tumor antibodies, that we'll discuss in a little bit, have opsonized a particular cancer cell. Now, in the previous slide, we just talked about how certain subtypes of CD4 plus cells can help augment the activity of the CD8 plus cells. Now, because those subtypes are primarily the Th1 subtype, it should come to no surprise that macrophages, specifically the M1 classically activated macrophages, are also involved in killing tumor cells, although some of the mechanisms aren't quite well understood. It's possible that they recognize the damage-associated molecular patterns put out by already killed tumor cells. They could also stimulate the secretion of IFN gamma by their Th1 subtypes that was just discussed previously, but otherwise we don't know too much more beyond these. Lastly, antibodies are certainly involved, and we know this because there's certainly increased expressions of antibodies whenever people have tumors. And we know that this is involved in a few different ways because we've just discussed them. So the antibody-dependent cell toxicity is involved in both natural killer cells and macrophages. And also in general, antibodies are used to activate complement, which themselves have some lysing capabilities, particularly for cancer cells. Although I will say this, that there is little evidence that any humoral immune responses against tumors have a significant effect in preventing the development or progression of tumors. So it's primarily CD8 plus cells with a little bit of help on the side. Now, unfortunately, sometimes the immune system just doesn't work on our side. And there's a lot of different ways in that the immune response promotes tumor growth. And we actually knew this well before some of these mechanisms were understood, in part because we knew that people with chronic inflammations or chronic inflammatory diseases were long recognized to have a risk for cancer. Now, some of the most direct tumor-promoting immune cells belong to cells within the innate immune system. Now, just as we learned that M1 macrophages are used to fight off against tumors, we almost want to think of their polar opposite, M2 macrophages, and also some other cells, as being tumor-promoting, in part because they're sources for VEGF, which is a growth factor that stimulates the formation of blood vessels and angiogenesis. So again, what's interesting about this is that a lot of times in biomaterials research, we want to hinder the M1 differentiation and promote M2 so that we can promote natural tissue regeneration. And it's almost the exact opposite in tumor growth. We want to promote M1 to fight off against cells and then hinder the differentiation of M2, which would proliferate tumor growth and cause for vascularization of tumor cells. Also, there are other innate immune cells and phagocytes that generate free radicals that themselves cause DNA damage and can initiate the start of the initial mutations for cancer. And then also many other cells like mast cells, neutrophils, and macrophages secrete soluble factors that promote cell cycle progression of tumor cells as well. Now, in addition to cells of the innate immune system, there's also cells and mediators of the adaptive immune system that help promote tumor growth. Now, I almost want you to have guessed this one, but just like M1 macrophages and Th1 subtypes of CD4 plus cells are used to fight off against tumors, we should already be thinking CD4 plus cells of the Th2 variety, in part because they're so closely related to the M2 related macrophages, just as Th1 cells are related to M1 macrophages. In addition, regulatory T cells, which function to inhibit CD8 plus cells and lymphocytes against attacking ideally pathogens, but also tumors, just allow tumor growth to continue unchecked. B cells may also contribute to the promotion of tumor growth to a certain degree by secreting factors that regulate tumor cell proliferation and also keep the innate immune cells chronically activated. Now, one of the hallmarks of cancer is the ability of cancer cells and tumor cells to evade attacks by the immune system. And so this is unsurprisingly a major focus of tumor immunology and engineering and design strategies for therapeutics. Now, tumor cell evasion strategies can be divided into two broad categories. The first one being what's called active inhibition. So this one we've already talked about a little bit before, in that PD-1 and CTLA-4 are inhibitory mechanisms, and these inhibitory mechanisms are often upregulated on tumor-infiltrating T cells. This results in a dysfunctional or exhausted phenotype of these cells because they no longer work. They're being inhibited by the tumor cells because these tumor cells have upregulated the expression of these ligands. Tumor cells themselves also secrete products like TGF-beta, which inhibit proliferation of lymphocytes and macrophages, preventing them from being killed. Furthermore, regulatory T cells suppress T cell responses against tumors. There's also a special class of suppressor cells called myeloid-derived suppressor cells. These aren't any one cell. In fact, they're a heterogeneous collection of many different cells and cell precursors found in tumor patients of those with chronic inflammatory diseases. So these suppressor cells work by secreting immunosuppressive cytokines like TGF-beta, IL-10. They also promote regulatory T cell differentiation, so they can help inhibit immune responses that way. A second, less direct way of evading mechanism, but also very effective, is the loss of the expression of the particular tumor antigens that are being targeted by CD8 plus cells. This happens through a process which is described as immunoediting. I like to describe it as like a natural selection because these cells are highly immunogenic. They have high genetic instability. So as they're rapidly proliferating and getting killed, ones that happen to have mutation that lacks the encoding for those tumor antigens, those neoantigens, are the ones that survive because they're not being marked to be killed. So if those new antigens that they're mutating are not required for the growth or maintenance of those tumor cells, they're going to result in rapid, unchecked growth, as opposed to those clones that lack that particular mutation, which are going to be targeted by the CD8 plus cells. The second form of this type of evasion has to do with the downregulation of processes related to class 1 MHCs. This includes both the direct downregulation of class 1 MHCs themselves and directly, or proteins required for the presentation of antigens through class 1 MHCs, such as the protein TAP, which we learned in the antigen presentation of T cells chapter. This in itself is also a natural selection process, as these cells get downregulated, or the cells that express the class 1 MHCs, they're targeted less by CD8 plus cells as opposed towards their clonal brothers that do express those and then do get targeted and killed by CD8 cells, leaving these downregulated ones the only lasting ones. And as we discussed before, that type of cells that downregulate the class 1 MHCs and the natural inhibitory mechanisms to natural killer cells, these types of cells are also prone to recognition by natural killer cells. As you can imagine, this leads to further downstream natural selection processes where these mutagenetic cells also acquire additional mutations that impair the expression of ligands needed for natural killer cell activation, in addition to the downregulation of class 1 MHCs. And so as soon as there's any other trick that the immune cells can somehow adapt to respond against them, these tumor cells mutate and then lose that. It's a big problem. Now, there are a number of different strategies for fighting off against tumors and cancer in general. Traditional therapies rely on chemotherapies and radiation, whose both primary effective means of working is to either kill dividing cells or block cell division in all cells, not just healthy cells. So it produces a lot of harmful side effects, as you can imagine. And in part, they're perhaps beneficial in early stages for cancers, but have been very unsuccessful in achieving benefits to cancers that have spread beyond their site of origin. Immunotherapies, on the other hand, aim to target the specificity of our natural immune system's ability to fight off against cancer. So it has the potential to be the most tumor-specific treatment that could be devised. This timeline discusses some of the new novel therapies that we're going to be talking about later on in this chapter, including monoclonal antibody therapies that target the inhibitory mechanisms, the checkpoint blockades, like for anti-CTLA4 and anti-PD-1. They also talk about the CAR T-cell therapies and some of their breakthroughs. Now, the idea of checkpoint blockades and the immunotherapy strategies against those, we've already talked about extensively in journal discussions and past lectures as well, but I'll touch on them again and give a quick refresher. But T cells have an innate checkpoint built into them through a number of different mechanisms to prevent any runaway immune responses. So it's in part to keep these in check. These include the CTLA4 and PD-1 mechanisms, and a blockade is a strategy of preventing these checkpoints from being carried out. So I always think of this as sort of like a double negative in that the checkpoint is being blocked, so there is no inhibitory mechanism. That is the functional goal of any of these checkpoint blockades, is to get rid of any inhibitory mechanism. And they're approved in part for first-line therapies for any metastasized tumors. As I said, again, chemotherapies and radiation treatments are quite ineffective against cancer cells that have spread from beyond their site of origin, so checkpoint blockades are one of the first things you go to whenever you have a metastasized tumor. They're the first-line therapy for those. Anti-CTLA4 antibodies bind to CTLA4, and they prevent them from binding and outcompeting for the B7 co-stimulatory ligand. So this targets naive CD8 plus cells. It prevents them from being activated in the first place because CD28 is needed to bind to B7 in order to activate, so these wouldn't be any good for already activated T cells. They're also necessary to prevent T regulatory T cells from caring about their function. Again, Tregs function primarily by just gobbling up B7 via CTLA4. Monoclonal antibodies against PD-1 or even the ligand PD-L1 are antibodies that prevent binding of any of those inhibitory mechanisms that are related to activated CD8 plus cells. So remember, CTLA4 is specific to naive CD8 plus cells. They prevent them from being activated in the first place, whereas PD-1 and PD-L1 are necessary to prevent runaway reactions of already activated CD8 plus cells. So these different therapeutic strategies work at different levels, and for that reason, a lot of combined therapies are currently being researched as an effective strategy to fight off against tumors. Now, unfortunately, there are a number of difficulties associated with checkpoint blockades and checkpoint inhibitor therapies. The most common adverse effects are just due to generalized autoimmune inflammatory reactions. As you can imagine, as we're turning off the inhibitory mechanisms, not just for the CD8 plus cells that are fighting off against our cancers and tumor cells, but any CD8 plus cell in general. So one common way of fighting this is to treat a patient with anti-inflammatory medications alongside the checkpoint blockades. However, there are a lot more specific difficulties associated with these checkpoint blockades. First of all, more than 50% of patients treated with either of these don't respond to the drugs or develop some sort of resistance after receiving an initial positive response. And there are a number of different mechanisms that result in this. The first one being that these therapies are unlikely to work against tumors with non-somatic mutations. That's in part because if they're non-somatic, then these neoantigens won't be carried over every time a cell reproduces itself. So you might only be fighting off one of the original lines of the cancer cells, but if they've already reproduced, then the CD8 plus cells that have their inhibition mechanisms inhibited or blocked aren't going to fight off against those new types of replicated cancer cells that don't have those mutations. Furthermore, in order for a tumor cell to be fought off by CD8 plus cells, Those tumors need a large number of what are called CTL tumor infiltrates, which is just as the name implies. It requires that CD8 plus cells are entering into the tumor and fighting from inside of it. It can't just simply be a fight on the surface, otherwise we're not going to get anywhere. So if a patient is demonstrating that their tumors have relatively little CTL tumor infiltrates, then it doesn't matter if we block the inhibitory mechanisms of those CD8 plus cells because they're not going to be able to do their job anyways. This also goes the same with patients that have high amounts of regulatory T cells. That simply means that our anti-CTLA4 or PD-1 strategies are going to have less an effect in those patients than ones with smaller amounts of T regulatory T cells. Furthermore, there are many tumors that don't take advantage of the PD-1 inhibitory mechanisms and use other immune evasion mechanisms. So in these cases, if tumors aren't inhibiting our CD8 plus cells, then it doesn't matter if we block their inhibitory mechanisms because that's going to have a little to no effect. And lastly, it might be the case that tumors have a high expression of the PD-L1 ligand, thus inhibiting CD8 plus cells, but as we kill those, they might develop or mutate into a resistant strain. Again, just through a natural selection process. The cells that end up surviving have smaller and lower expressions of the PD-1L ligand. So again, it doesn't matter if we inhibit that checkpoint system if they don't express that ligand or express less of it later on. There are also strategies for what are called tumor vaccinations, which is a vaccination of tumor-bearing individuals with tumor antigens as a way of stimulating CD8 cell responses against those particular antigens. Now within that, or maybe alongside that, there are a few different strategies that can belong to this sort of broad category. It could be simply stimulating nonspecific immune stimulations, like through adjuvants. It could be the injection of already killed tumor cells that would have those neoantigens. It could be more sophisticated mechanisms like injecting recumbent tumor antigens that would then be picked up and recognized to stimulate CD8 plus cells. Or it could be the injection of patient-specific dendritic cells that were then incubated with those tumor antigens. So in this case, this isn't exactly adoptive cell therapy because you are taking the host cell responses and it's not their own lymphocytes that you're taking, but it's the antigen-presenting cells that you're taking and making them more effective at presenting those antigens to your own CD8 plus cells. Now, most of these strategies aren't true preventative strategies. They're what are called therapeutic vaccines because in all cases, the host has already developed tumors. So we're not vaccinating hoping that a person won't develop more tumors. It's more of a way of fighting off against the tumors that a patient has already developed. However, there are true preventative strategies that typically target viruses that are linked to cancer, like HPV or the Epstein-Barr virus. There's also cool strategies in the development of DNA vaccines and viral vectors that encode tumor antigens without our own host cells directly. What's great about this is that because they're viral vectors or they work on DNA vaccines, these neoantigens are directly coded within the cytosol of the cells, which presents them directly to class 1 MHCs. So it's a very direct and efficient mechanism for presenting these neoantigens to our CD8 plus cells. Now, these all seem like really cool strategies. I hate to end this slide on a downer, but overall, the results from trials of these different types of tumor vaccines has been pretty inconsistent, and none of these are considered successful at all. But I'll try to end this on an optimistic note and say that hopefully the new generation of engineers are capable of engineering strategies that use these that are more successful in ways in which they haven't been so in the past. Good luck to everyone, and credit me when you do it. Thanks. Now, one cool strategy that we've talked before in class and has been really one of the new breakthroughs in the past 2 or 3 years are what are called CAR T cells. Now, the CAR stands for chimeric antigen receptor, and these are receptors that are genetically engineered with tumor antigen-specific binding sites on T cells, but they have a couple other cool things about them that make them unique. First of all, they're encoded by recumbent immunoglobulin variable genes. So these are a lot more like a membrane-bound antibody on B cells as opposed to a T cell receptor found on T cells. But why stop there? Because engineers thought, well, why don't we put in these cytoplasmic tails that contain all the signaling domains necessary to activate a T cell. So not just portions for the T cell receptor, but also the co-stimulators like CD28. That way we make this all-in-one package receptor as opposed to needing a number of different things in addition to the T cell receptor like we do with classical CD8 plus cells. Now, one of the coolest reasons for using these antibody-type receptors as opposed to T-cell receptors is that it eliminates the MHC class restriction of T-cell receptors. MHCs are genetically encoded, so again, you can't just transfer lymphocytes from one patient into another because they're not going to recognize the antigens displayed on someone else's MHCs. It's not of the same genetic trait. However, antibodies have no such restriction. This allows scientists to make sort of a generalized receptor that are sort of like a one-size-fits-all for all patients. They're not patient-specific. They're not genetic-specific. The general protocol for creating CAR T cells is that they'll first draw lymphocytes from a patient and isolate their T cells. They'll grow and expand them in vitro using a number of different antibodies and cytokines like anti-CD3, because CD3 needs to be triggered in order for a T cell to activate. Anti-CD28, because CD28 is a co-stimulator and it also needs to be activated in order for that T cell to proliferate. And then IL-2, which is a T cell growth hormone. So all those things allow a patient's T cells to be expanded to the point where they have a large enough pool to fight off against what they need to. They transduce the CAR gene via retroviral or lentiviral vectors, which then induces the chimeric antigen receptor on those particular T cells. At that point, they transfer those lymphocytes that they've grown in the lab back to the patient, and then the patient's own cells that are now coated with the CAR receptors fight off against cancer. Now, as excited as the world is about CAR T cells, there are a number of significant roadblocks that we have to get over in order to make these truly effective treatment methods. The first is what's called the cytokine release syndrome, also described as a cytokine release storm. Because we're transfecting so many activated T cells that are activated at once, they all release a large amount of cytokines which cause an intense systemic inflammatory response. Because of this, patients are often co-treated with anti-IL-6 antibody. IL-6 is an inflammatory cytokine that can help reduce this cytokine release storm. Furthermore, surviving tumor cells may lose the antigen that's actually being targeted. At that point, we have all these transfected, very expensive T cells in the body that aren't able to fight off anything because they've killed off everything that's been expressing that neoantigen, yet cancer still survives. This is another similar natural selection process due to the highly mutagenetic and proliferative capacity of cancer cells. And just like T cells become exhausted and unresponsive over time, CAR T cells can become responsive over time. There's a number of different potential strategies that can overcome this, such as genome editing that can remove the PD-1 receptor, the inhibitory receptor on these T cells. Or while we're at it, let's just eliminate all endogenous T cell receptors entirely. That reduces autoimmunity and eliminates the checkpoint inhibitor mechanism, which is downstream and linked to PD-1 and CTLA-4 in general. We can also try and induce memory-type qualities to T cells, such as switching their effector mechanisms from CTLA-4 to effector memory cells that allow for both lasting memory of these types against these types of antigens, but also rapid proliferative capacity whenever that antigen is then reintroduced or found again by these cells. Also, so far CAR T cells are only approved for blood-borne cancer types. So there's been so far no success with solid tumors. So we have poor CTL infiltrates with CAR T cells. Other strategies include passive antibody therapy, which involves the transfer of tumor-specific antibodies into patients, which is a rapid and theoretically a very specific approach, but does not lead to long-lived immunity. Again, that's what makes it passive as opposed to adoptive. Some monoclonal antibodies have been in use to treat cancers for over 20 years, and many more now are being approved or in advanced developmental stages. I should also note here that checkpoint inhibitors, while they are antibodies, do not themselves bind to tumor cells and thus operate on entirely different mechanisms from what we're talking about here. Now, there are a number of different strategies in which these passive immunotherapies work. The first being that they can really just opsonize tumor cells, and then by binding to the cell surface of those tumors, they can engage in host effector mechanisms through the Fc portion of that antibody that results in killing of the tumor cell. There are a couple of different ways that this can come about. This could be through natural killer cell-mediated cytotoxicity, like through anti-CD20 that's approved for B-cell lymphoma. They could initiate complement-mediated lysis. They could also utilize complement or Fc receptor-mediated phagocytosis via macrophages. Another strategy relies on these antibodies attaching to or blocking specific cellular functions that interfere with the signaling or proliferative capacity required for tumor growth. When I first talked about oncogenes, I mentioned the different growth factor receptors that are overexpressed in some cancer cells. well, then we can have antibodies that target those receptors, and because they're overexpressing these particular receptors, they're going to cause for a lack of rapid proliferation in cancer cells more so than in healthy cells. So that's exactly what the strategy is for the anti-HER2 antibodies that attach to the HER2 growth factor signaling receptor overexpressed in breast cancer cells. There's also the really novel bispecific T-cell engagers, which I like to call BiTEs, that are recombinant antibodies engineered to express two different antigen binding sites. Remember, antibodies can bind to two separate things. They have that Y-shaped. In this case, these antibodies are engineered to have two different binding sites. One would be for a tumor antigen, and another would be for a T-cell surface, most commonly CD3. And these are thought to form sort of an immune synapse that's brought about by cross-linking these tumor cells with the T cells, and thus allowing them to produce their jobs at a more direct and localized site. Lastly, there's a way of targeting immunotoxins in a much more specific way by the use of antibodies. So this would rely on conjugating certain chemotherapy or radioisotope drugs to monoclonal antibodies that are specific to neoantigens. And that way we can deliver those chemotherapeutics in a much more direct and targeted fashion. Other types of immunotherapies include the delivery of cytokines that induce the proliferation and differentiation of T lymphocytes and natural killer cells. These include the T cell growth factor IL-2, which is approved for therapy against a few different cancers. There's the treatment of IFN gamma, which is approved treatment for several cancers in addition to other diseases. Again, these increase the cytotoxicity of natural killer cells. They increase the expression of class 1 MHCs necessary to present neoantigens, and they also increase the ability of dendritic cells to do their job. They're the most effective antigen-presenting cells there. Lastly, there's the delivery of growth hormones for non-lymphocytes that are used to increase leukocyte proliferation, particularly after chemotherapies and radiation therapies. There's also the local administration of non-specific inflammatory stimuli, which really just functions as a polyclonal activator of all lymphocytes. So these are ones not specific to just the CD8 plus cells targeting tumor cells, but all lymphocytes in general. So think of this as just a large injection of adjuvants. Lastly, there's the cool therapy known as graft versus leukemia effects, in which case leukemia patients are administered with T cells and natural killer cells from another patient, along with allogenic hemopoietic stem cells that in all can contribute to the eradication of tumors and then the replenishment of leukocytes. So the administered T cells target host leukemia and HSC cells, so thus they destroy almost all leukocytes. The natural killer cells would target cells with downregulated class 1 MHCs, and then the allogenic HSCs help replenish all the lost leukocytes that were eliminated by those T and natural killer cells. So in summary, I think in this chapter we learned about a lot of the different mechanisms that can result in cancer, the ways the body fights off against cancer using natural mechanisms within the innate and adaptive immune system. And then on top of that, ways that cancer has developed to evade the immune system. And then lastly, strategies that engineers have developed to fight off against cancer, using everything that we've sort of developed in the preceding chapters up until now. So as always, thanks for listening.